Hi, this is Miss Litton, and this is 7th period review for AP Biology, Nutrition, and Transport in Plants. Say hi.
Hi.
All right. So, first thing, we want to know this, okay? 95% of the dry weight, carbon, hydrogen, oxygen, and you want to know where does it get that? How does it acquire it? Okay, could you write an essay on how it acquires what it needs? So how does it acquire the carbon that it needs?
Oh, it comes through
Stomata, right? Through the stomata. That's how it gets the carbon that it needs. Okay, how does it get the hydrogen that it needs? Water, and that comes through the roots. How does it get the oxygen that it needs?
Roots. It's from the roots. So it does photosynthesis and then it makes it comes into the
Stomata, oxygen. Hi. Right? Don't forget about that one. Yes, you have oxygen when you have water, so you can get it from there. But don't forget about just straight up oxygen, right? Remember how we use that? Yeah. What process do plants have to do?
photosynthesis.
And
Respiration.
there we go. Yes. Okay, and then all the other nutrients in through the roots. And we said there's all different kinds, and we talked about what a mineral is, an inorganic substance. You don't need to know all of them. We talked that it has a nutritional role. There's no substitutes. It causes a plant to die. We looked at this as possibly a supportive statement when you're trying to remember. See Hopkins Cafe, managed by my cousin Clyde Moe, to remember the important nutrients that a plant needs. We talked about how you could identify that by hydroponic studies. What are hydroponic studies? Using just what? Water. And you have all the essential nutrients you need dissolved in the water except for one. Yeah. One, and then you see how that impacts the plant growth. Could that be a quantifiable study? Could you get data? Yeah, how much your plant grows. We looked at what soil is composed of, and when I look at that 25% air, why is it significant that soil has air? You know, a quarter of it has air. Why is that significant?
So
I'm sorry, I don't hear anything. What? Yes.
Could it acquire some air through the roots of the plant?
Yeah, why does it need to acquire air through the roots of the plant? For cellular respiration. Yes, because roots are not doing photosynthesis, they're just doing cellular respiration, right? And they need oxygen in order to do that. And if your soil is too compact, if your water is not getting turned over, if it's stagnant, it won't have enough oxygen in it. If your roots are in clay with tiny, tiny little particles that don't leave space between them, you won't have the oxygen that you need. Okay, obviously you gotta have water. You need minerals. We talked about that, and organic matter is in the soil as well. All right, we looked at particle size. This has a lot to do with sustainability of that plant for you to have spaces. If you have all large particles like sand, the spaces are so big that the water will go straight through and it won't be able to hold water. If they're all small, it won't be able to hold air. You want it to be just right. And just right is usually referred to as loam, is the perfect particle size. Okay. And then once we learned about that, we talked about how things go in through the roots. What is the outermost layer of a plant called? Starts with an E. Outermost layer? Epidermis. Good. I would just say as a strategy when you hear endo that means like inside. Endodermis, epidermis would be on the outside. So the on a mature root, if the zone of maturation, there's the zone of cell division, zone of elongation, and zone of maturation, there are these root hairs. Root hairs do what? Why is that a great adaptation? They increase surface area for a greater absorption of both water and minerals. So we know that plants, we know about cell membranes because we've studied them extensively, and we know they're a phospholipid bilayer, and we know that if you are large or charged, you're not crossing. So we need to figure out a way to make that happen. And so we talked about, and we revisit it multiple times. There's a pump. What do we call it? Proton pump. Yeah. Pump hydrogens out in exchange for something like potassium to come in. Or do a co-transport. Pump hydrogen out, who's positively charged, hook them up with some negative ion, and they go in all neutral. Okay? And once those molecules move in, we talked about ways things can move through the plant. I'm just going to jump to that, and then I'll come back to those other pictures. We talked about the apoplast and the symplast. Okay? Which one goes between the plant cells? Apoplast. Okay? And if you go from one living cell through the plasmodesmata to the next living cell to the next living cell, that is the symplast. Which is more highly regulated? Symplast. Which channel gets shut down eventually? Apoplast. And who does the shutting down?
The Casparian strips.
Casparian strips, which are wound around the endodermis. And then that forces water to be regulated. So we know water moves. There's no water pumps. There are things that can facilitate water moving across the membrane, like aquaporin, but there are no water pumps. So if we want to move water, we got to motivate it. How do we motivate water to move? Create a what kind of environment? Hypertonic environment, because water must flow from the hypos. So how can we do this? We can pump minerals into the stele, the centermost part of the root. The stele is anything inside the endodermis. Pump those in there, and water will flow from a higher water potential in the soil to a lower water potential in the cortex of the root to the lowest water potential in the stele, and then it's going to move up the xylem. Now, because it can't go back out. And we see evidence, you don't even need transpiration to get water to start moving up the root and the root stem. What do you call that? It's called something pressure. Root pressure. Root pressure is caused by minerals getting pumped into the stele and water flowing in, and it can't flow back out. It can't flow back out because of the regulatory process of the endodermis, so it can only flow up. Okay, and that's why a stump bleeds when you cut it. Okay, now that's not enough though. Root pressure cannot overcome gravity. We need something more powerful to move it all the way to the tippy top of the plant, and that's when we talk about the TATC theory, right? And let's talk about what those letters stand for. T, the most important of all those letters is transpiration, right? So water evaporating out the open stomata. Why are they evaporating? Because they're going from a higher water potential to a lower. What could facilitate their evaporation? You're doing a lab right now. What could facilitate it? Wind, solar, sun, right? Those types of things can facilitate transpiration. And when a water molecule evaporates, it never evaporates alone, because it's always connected to another water molecule through what process? Cohesion. And then it's supported to the xylem wall via adhesion. And then what was the other T? Tension. Okay? Tension of it going from a higher water potential to a lower water potential. It's always lower outside, and so it keeps getting pulled and pulled. Making water move is all about the pull. So going back here, A18 number. Don't forget to find a class. There's more than 5 of you. You should all be logging in. There's 21 of you. Why am I only seeing 5 and not 21? Because what? Huh? 21 of you, not 12.
Look for the answers different from all the others. Sunday right there.
You think so?
So there's like essential inorganic.
We're weighing in.
Tell yourself.
21% of you passed. Good job, Zach Efron, Jeep Wrangler, and PETA, Malark, on top. Why is the answer nitrogen gas?
Is it just nitrogen fixation?
Yeah, it can't use nitrogen gas at all. It needs to be in a symbiotic relationship with who? Something like cyanobacteria found in your root nodules, right? That fix nitrogen gas for you. Okay, so that one was not a nutrient for plants. All the rest were. Okay, tell your bio buddy, were you one of the top 3 or not so much?
Johnny, you got it wrong.
Are you kidding me? You told me.
Which is,
You can try this one. It's okay.
well, it's freaking out. What's it's part of the, it's like a raised milk part of it.
It's on like something.
Pretty confident.
Wait, no, it didn't. She
Whoops. Forgot to mark her. Yay!
I see me. I'm at the bottom. What are you?
All right. So we talked about this, apoplast and symplast, and we talked about this already. Why doesn't the person in the gold seat just do a rundown of this page right here? This is a good page. Go ahead, gold seated one.
Gold seed.
Oh, darn it. Man, that was a throwback. Blue. I used to have gold and brown.
That's cool.
Goldies and brownies is what I used to go. Man, I haven't had gold seats in like 3 years. I don't even know where I pulled that one out of. Blues. Go Blues.
Facilitating, it's not natural. It's cooking. It requires work.
Fixation. Yeah. So it comes out tomatoes. But also.
All right. Are you good on this? Okay. So we talked already about nitrogen-fixing bacteria in the root nodules. We talked about mycorrhizae association with a fungus to increase some mineral uptake and definitely water absorption. Both of these bacteria and fungi are paid back with sugars. And we don't need to know about parasitic plants. Ooh, okay, so xylem and phloem. Xylem, there are 2 ones you really need to know. So do you remember the names of the 2 before I showed you?
Vessels and tracheids.
Yeah, tracheids and vessels. You could have a vessel on a tray, like a cup on a tray, vessel on a tray, kids. Okay, the tracheids are smaller. Vessels, think vessels, pipes, bigger. And then with phloem, they consist of 2 parts. What are the 2 parts? Sieve tube and companion. Who needs to be alive to work? Phloem. In fact, xylem works best when it is dead. Okay, because then it's just a full-on pipe that water can move through. Remember, it's based on transpiration. If you have an open pipe, then it can actually be more effective. You better get a hundy on this. If you're in here, there are a couple teams who don't have their plants sitting in cups. I would get a cup and make sure your plant is sitting in a cup to protect it.
You're going to chosen APBio. We've been there.
Ah!
How did we miss that?
What?
Yeah. Okay. Good job, relationship goals.
¿Por qué?
Okay.
¿Dónde está?
This is evidence of root pressure. And we have gone through, I already went through the discussion with you about xylem, so I feel comfortable about that. So let's move on to stomata, how they open and close. Okay? Again, let's talk, we're revisiting something we talked about before. Who is that? Proton pump. So if I want the cells that create the opening of the stomata, those are called what kind of cells? Hard cells. And I want them to expand. Okay? And so if I want them to expand, what ion do I need to get in there? Potassium. So that's a positive ion I want to go in, so I better put a positive ion out. So what am I going to use?
Hydrogen.
Called the, yes, and I'm going to use the proton pump. Thank you. Pump out hydrogen, potassium comes in its place. Who follows the potassium? Water. Perfect. And yes, agua. So that will cause them to swell. Conversely, it'll make them go flaccid if you're not continuing to put that potassium back in. Next, let's talk about phloem. And here you can see an aphid tapping into the phloem, and the phloem's coming in so strong, it's actually coming out its cloaca. Okay? And yeah. Okay? And it's called the pressure flow hypothesis. Whereas the way xylem works is with a pull, phloem works with a push. And so basically what you need to do is take your sugar, concentrate it in your cells, get water to follow it in, and that increases the turgor pressure, right? And you get it going from your source to your what? The area where the sugars come from is called the source. Where they're going, getting consumed or stored, it's called the sink. Exactly, source to sink. Can the source and sink change? Well, yes, they can, Miss Litton. Because in the springtime when you have green leaves, it's the leaves that are the source generating the most sugar. But in the wintertime when the leaves have fallen off, now the source is where you stored those sugars in the roots. So the roots, which were once the sink, become the source. Growing new shoots and leaves would be what? Brand new shoots and leaves and stems. Would they be a source or a sink? Sink, because you're growing. Okay. So leaves produce sugar, transport them into the phloem, water follows, turgor pressure increases, push it to where the sink is. Because you're always going to be going from a higher concentration of sugar to a lower concentration of sugar, because you're going to pull the sugar out and store it. And when you store it, it's not individual sugar molecules again. So you can keep getting it to go from high to low.
First, guaranteed.
How do we get the sugar to go where we want it to go again? I didn't mention this before. We have to use the what again? Proton pump. Good.
Okay.
Sure, check that. Yay!
You get a car shirt. I started tutoring.
Can water ever follow by active transport? No. Can you ever actively transport water? No. You can actively transport other things and water will want to follow them. But water we cannot pump. Okay. Now, let's talk about lab questions you might have. Okay? So ask away. Ask her. Ask her. Yes.
I think
asked you already, but I don't know. For the analysis, is it one analysis or four for each gene?
One analysis. Your ultimate analysis, you're looking at all these genes and you're determining what organism or type of organism it evolved from. What are you guys starting to think? Did it evolve from a fly? Is it closer related? Now, does it happen to share a similar gene to a fly? That particular gene, is it the same as a fly? And could it be the same in us as well? Sure. It could be one of those conserved genes that we all have. A housekeeping gene that all of our, like to code for a cytochrome in mitochondria, right? For a universal process like cellular respiration. So we would all have that same gene. Okay, so then you're looking at the other ones. What are you starting to think that what type of organism are you putting it more closely to? Some sort of bird. So you've got to support that using those genes, the gene data. Yes?
You said that you didn't have to go through, like on the blast lab, like when you were doing it on the board, you were saying that just put it in blue and don't put it in paragraph.
Like for each number, just answer below.
Yeah, answer below each number. Yeah, just do that. Yeah, I like that. That's easier to
I
grade.
said it in the review.
I said in the review too. Okay, I'm glad. Okay, good.
I
All
can do
right.
every lab like that. I feel like it's easier for you, and it's easier for us.
Because I have to push you to think.
We think.
Right.
We're thinking.
Yes.
I've got a question about the transpiration pre-lab due tomorrow.
Yes.
Is this done in the lab notebook on a separate sheet of paper?
The only reason I would say put it in the lab notebook is so you don't have to redo it again. But if you want to put it on a piece of paper first, go ahead. Whatever you feel comfortable with. I'm trying to ease your load. I'm trying to be your samwise ganji. Carry your
What does that mean, guys?
You know what that
Lord
means?
of the Rings, Santa
Lord of the
up the
Rings.
mountain.
I haven't watched.
Let me share the load.
That wasn't Hindi. Okay. That wasn't Hindi.
But yeah, Frodo had to carry it alone. That's just the way it was. Yes.
For the blast lab hypothesis, are we making a prediction about what organism is going to be descended from,
Or the process itself.
or the process?
Yeah, so at that point, you're making a hypothesis that you could identify. It's more along the process, that you could use something like a DNA to do that, okay? Now, did you make a hypothesis? You can't make a hypothesis about what kind of organism it is until you actually analyze the data. So you can't make a hypothesis like, I think it's a turtle, because it looks like a turtle, okay? No, you're making a hypothesis that you'll be able to place it on that cladogram based on similarity in genes. Okay, what else?
On its directions for the hypothesis, doesn't it say to put the organism on the diagram where you think it will go?
Oh, your initial one, by the way it looks?
Yeah.
Yeah.
Does.
Using the
So
morphological
did you
traits.
do a hypothesis like that?
Yeah, I wrote mine about. what
Okay,
the
I won't
hypothesis
worry. You can do it either way. I'm happy either way.
Okay.
Don't change anything. Don't change it. You know, if you've written it and it's good, I'm good. Okay? I won't mark you off. Remind me later. It's recorded for all time. You could say, miss what you said. What else?
I really wish it was not.
Donde?
Chapter.
Come on.
A specific organism that it matched most with? Do we say that, or do we say?
Gallus, perhaps?
No.
No, it's like you're putting it in a category of organisms.
So would it be?
No.
Do you understand?
What else? Anything else? All right. What do you, yes.
What was terms in tomorrow?
27.
I can't hear. Shh.
The hypothesis for the transpiration, is that just variables or just arms?
You're gonna make, so your best hypothesis would include all the variables. Your best hypothesis includes all the variables, because you're gonna say, I think transpiration will increase under these conditions and decrease under these conditions because, whatever. Yes?
So regarding the confusion,
We're back to the blast lab.
Yeah,
Okay.
back to the blast lab. So we're not really sure exactly what to analyze in our conclusion. Are we analyzing just like each organism individually and comparing it to the
No, what you're doing there is you're taking your organism, your unknown fossil, and your analysis is based on this gene and this gene, you know, and I'm going to assume this gene is blah, blah, blah. I'm going to assume it's this organism. You're making a claim about what organism you think it is. Here's my evidence. Okay, this is why I think it's this, because you can compare a lot of genes, right? And you could compare, I think, didn't your final have a question where I gave you, like, where it compared one gene and they were all over the place. You couldn't make any groupings. Wasn't with the cichlid and everything. Like you couldn't make any connection by one of the if you organized it this way. But if you looked at this other gene, you could group it together. The high plate did you have a platedness one?
Yeah, it was something about like
Yeah, remember? It was like one of the massive ones. Like read a paragraph, study 5 diagrams, and answer these 10 questions. And there was one like that. Yeah, so you could look at a lot of different aspects. You have to rule out like which do you think is important, what you know, to distinguish between when you look at all the genes that you had to look at. What else?
Just re-clarifying for the analysis, you want us to pick one specific gene.
It's not that you're picking one gene. You're picking an organism and supporting it with the genes data, right? What you think it's going to be.
So if I wanted to prove that it was more of a bird, and then I showed that 2 of the genes were highly close to a bird, I can just do that.
Right. But you're making a claim that I think it's a bird.
Yeah, and then also my conclusion, can I talk back to the original placing of the cladogram based on the morphological stuff and compare it to how I would change it?
Yes,
with the DNA
sure.
Is that fine?
Yeah.
It's
Talk back to your earlier self.
Yeah, it's pterodactyl.
like,
Yeah.
It's a pterodactyl. It's a bird that.
I can't hear. What?
If we think that it's one organism, it's actually like a different one, are we gonna get graded off on that?
What do you think it is?
Either an alligator or a chicken.
Yeah, that's what I was.
But I was like,
Are alligators and chickens connected?
they're down the same.
Vertebrates.
Yeah, so think about that. So go along that. Yes.
In our analysis, when we put like a picture of like our background after we do the
Yes.
and everything, do we have to, since it's on a question, do you want us to justify like the reasons why we put it there?
Your evidence reason justifies why you put it there. Yeah, but you're in.
But in there, there's.
Yeah, in there you're justifying its placement.
Oh.
Yeah. What else? Yes.
So even though, like, let's say for gene 1, like it says it's a certain kind of chicken or like a certain kind of bird or something, we can just say that we believe it is some kind of bird, right? We don't
Yeah.
need to say what kind of bird.
Yeah, you don't know what kind of bird.
Japanese quail.
He thinks it's an alligator.
I think it's a T-Rex. It's a T-Rex. T-Rex is a chicken shape that is plating of an alligator. Okay.
I'm still reviewing. I'm still recording.
Pterodactyl fly. It's a pterodactyl.
Okay, anything else? Can I stop this recording? Wait, let me hear your question.
Are we having a shadow?
Oh, yeah, good question. So, did I say we were going to have a shadow?
No.
Okay, so because what I was thinking is you have a lab to prepare for, you have a lab to turn in, and you have terms, and you have a quiz. That doesn't sound to me shadowlicious-y. I don't think you want a shadow when you have all of that to do. So no shadow. I just think it's too much. No, okay. Yeah. No shadow. No shadow. Have a piece of toast. Don't stay up too late.